As in all things, shape dictates function. That's what we're going to be looking at here when we are talking proteins for this short mini lecture. Now, shape determines function of these proteins, and it's such a detrimental determination of the correct shape to get the correct function that we have examined and have set 4 levels for protein structure. First level being primary. Primary is going to be a unique sequence of amino acids. Amino acid 1 followed by the second, the third, the fourth, the fifth. The secondary structure is going to be composed of the coils and any folds that we'll see in this straight chain of amino acids. Remember, the amino acids have a 3-dimensional shape. So they're going to be pushing and pulling against each other because of their overall size and their shape. Well, that pushing and pulling against each other can form a coil. It can form folds. It can form straight parts, this parts. And we're going to see. Now, if they're forming, in certain parts they're forming coils, other parts are forming folds, well, what are the sequence of those, coils and folds and twists and turns? That is what we call the tertiary structure. Tertiary structure is nothing more than helix, turn, straight part, turn, helix, helix, turn, blah, 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 until you hit the end. And then quaternary structure. How many polypeptides have to be there? 1 polypeptide correctly formed, quaternary structure set, you have a functioning protein. Or does it take 2, 3, 4, 10 of these polypeptides coming together before you have a complete protein, the correct quaternary structure, before you have a functioning protein? So let's start at the bargain basement. Primary structure. The sequence of 1, 2, 3, 4, 5, on through to the end of the amino acids. This is going to be set by your genes. The genes will say this is the sequence. They will be linked together using a dehydration reaction like we've talked about before, and that gives you the primary structure. Now, as I said, the amino acids, each individual has a specific shape, and its shapes are pushing and pulling and crowding against each other, and they will induce this spiral, a corkscrew shape they refer to as an alpha helix. Or they can equally push and pull against each other, and they can form what they call a beta strand or pleated sheets. Your sequence, glycine, proline, threonine, as you see here, have 3-dimensional shapes. How the base molecules are pushing and pulling against each other will give you the alpha helix, will give you the beta strand. The sequence of helices, strands, turns in between and whatnot, that's the tertiary structure. The tertiary structure utilizes hydrogen bonds, ionic bonds, hydrophobic interactions, and that weird van der Waals interactions to hold everything together. Most importantly, they're going to use what are referred to as disulfide bridges. So you look here. Here's the amino acid sequence, just showing you the secondary structure of an alpha helix. Within there, the R groups from the individual amino acids are going to interact with R groups from amino acids in other areas. They're going to form hydrogen bonds. These hydrophobic interactions are going to extrude water. Ionic, opposites attracting. But what holds everything together the most is right here. Disulfide bridges. Sulfur in the R group from one goes to a sulfhydryl in the other, and you get this nice, strong, covalent bond. These other, ionic, hydrophobic, hydrogen, these are extremely weak. Remember, easy to make, easy to break. Individually, they don't amount to much. You get enough of them together, you might have something. But this disulfide bond, that's strong, that will hold the protein together. But then the question comes, how many polypeptides do you need? Is it one polypeptide's long enough, big enough to have a function all on its own? That's the quaternary structure for that protein. If it turns out, though, that polypeptide is only a small part, and you need more parts to get the complete protein, then you see something here. This example, transthyretin polypeptide, you can see here, this beta strand here, this alpha helix, showing you examples of secondary structure. You start here, you end here. You go through beta, twist, turn, beta, twist, turn, alpha helix, twist, turn, back and forth, till you end there. Tertiary structure. But this polypeptide, all on its own, doesn't do anything. The polypeptide has to be with with 3 others. A total of 4. 1, 2, 3, 4. Then you get the complete protein that has a function. Another example, hemoglobin. Hemoglobin has 2 alphas and 2 beta polypeptides. The 2 alphas are the same. The 2 betas are the same. Individually, they do nothing. 2 or 3 out of the 4 won't work. You need all 4 in the correct shape. Great example of what happens when one of these levels of structure is interfered with. Sickle-cell disease. Sickle-cell disease is an inherited blood disorder where the red blood cells, instead of being nice and round, have a sickle or a crescent moon shape. Because of that weird shape, they get caught up in capillary beds. These individuals are prone to bruising, bleeding. Because of the abnormal shape of the red blood cells, abnormal function of the hemoglobin in those blood cells, their blood oxygen levels are greatly reduced. It means they're going to have physical developmental issues. They may even have mental developmental issues. Start at the primary. Here we see primary structure. Primary should be valine, histine, leucine, threonine, proline, glutamine. This gives you a nice beta subunit, which associates 2 betas to 2 alphas, and gives you free-flowing hemoglobin that free floats within the red blood cell. Nice, round, dimpled like it's supposed to be. This mutation takes that glutamine and replaces it with valine, 1 amino acid chain. Gives you this abnormally shaped beta subunit. Incorrect shape, but it still associates 2 betas to 2 alphas. But now, instead of being this free-floating hemoglobin, this hemoglobin has a greater affinity for itself. The beta subunits start to glom onto each other, and they start to form larger and larger crystals that stretch and distort the shape of the red blood cell. This cannot carry oxygen very well. This gets caught up in capillary beds. This does all sorts of weird things. Now, once the shape of the protein has been set, the polypeptide, it's not permanent. Things can interfere with that. Remember, it's being held together by ionic bonds, hydrogen bonds, so anything that interferes with those will interfere with structure. I mean, look at it. pH. pH is nothing more than free hydrogens. Add in enough free hydrogens, you interfere with hydrogen bonds. Same with salts. Add in enough chloride, you're going to interfere with ionic. Temperature, well, that's just cooking. You cook an egg, you denature it. It goes from clear, the white's going clear, to opaque white. What you're seeing is the denaturation. The proteins literally are falling apart. They're unraveling. You're breaking the disulfide bonds. Denatured protein is nothing more than a protein that has the primary, maybe the secondary structure. It will fold, get in the correct normal shape. You add in free hydrogens, lower the pH, increase the temperature, something like that. You're going to go from a normal folded protein, something unfolded. Many proteins can go back. All depends on how many of those disulfide bonds are broken. Now, sometimes getting proteins to their correct shape, the polypeptide, to their correct shape, may be a bit difficult. So, there are proteins, complexes, known as chaperonins, that will go and will help polypeptides get to the correct shape, the correct folding, the correct twists and turns. The lack of these chaperonins is what we think leads to Alzheimer's, Parkinson's, mad cow disease. A hallmark of these three diseases is the fact that the neurons in the brain start to accumulate misfolded proteins. Can't fix them, can't get rid of them. What you have, the chaperonin, is a collection of proteins that opens up and a polypeptide enters in. Polypeptide goes in, cap goes back on, and the proteins in here push and pull and then fold everything correctly.